Before we go to the Q&A session, and I'll remind our guests again, please do submit your questions. I'd like to welcome back Christian from our earlier sessions, both day 1 and day 2. And also I'd like to welcome Janeta from Rectify Pharmaceuticals. Thanks to all of you for joining the discussion session today. Perhaps we can start with you, Janeta, and perhaps you'd like to introduce yourself, tell us a little bit about Rectify, if you like, and also share your thoughts on the application of chemistry automation, perhaps where this has proved useful to you in your career.
Yes, absolutely. I do appreciate the opportunity to participate in this panel. I'm a med chemist by training. I have had the privilege to work in Cambridge, Massachusetts biotech for about 20 years now. This is my 4th startup. So I started back in industry after completing my PhD at Dartmouth College. I studied at a small biotech named Neogenesis. Neogenesis was a platform company and had the automated ligand identification system, an ASMS-based mass spectrometry platform for screening targets that many folks in industry may be familiar with. And later in a few years, long story short, Neogenesis became Schering-Plough Cambridge site, and then it became Merck. So after about 10 years in the same place, let's say a satellite site where we maintained the biotech spirit, kept it to a small site of about 100 to 150 scientists, I made a transition to an even earlier biotech that was Agios Pharmaceuticals, that was focused on cancer metabolism in oncology and rare inborn error of metabolism. So Agios functioned in and this will become relevant to why we use, let's say, organizations like WuXi that actually enable and facilitate our research. Was a hybrid model, both internal research, but also a lot of outsourced, functional discovery resources. I was there for about 6.5 years, and then I transitioned to my 3rd biotech, which was Decibel Therapeutics. That's in a very, let's say, difficult therapeutic space of hearing. We don't have, as an industry, therapeutics hearing yet. We are on the way. That has been a much, let's say, a little bit more challenging area to drug and make an impact. I'm here at Rectify. We are a seed company that is funded by Atlas Ventures. We are still in stealth mode, but we are dedicating our efforts to rare genetic disorders. And we are studying programs on small molecular efforts to do so. All the way throughout, let's say, the programs that I have called, from hits to enabling delivery of clinical candidates to patients, it's very important to be effective and execute. And, it's very important, as the speaker said today, how do you enable those hits to be able to progress them fast and get them to reality. And in my experience, having worked with, let's say, library design teams and making, effective progress, that has been tremendous in, let's say, enabling the way we can push discovery from key stages that you need to push molecules towards the drug candidate, and that's hit to lead and lead optimization. How quickly you can enable parallel synthesis to explore certain areas of the molecule, to understand how you build in properties. In my experience, and I've been honored to work with exceptional teams, that has enabled the speed of the drug discovery process. That's what we need to do as drug hunters, be able to execute efficiently and push molecules in the clinic as fast and as effectively as we can.
Fantastic. Thanks a lot, Janeta. Perhaps a general question to the group to consider where in the various stages of a drug discovery program, and laid out, a pipeline, a typical sort of drug discovery pipeline. We've spoken over the last couple of days about virtual screening, which is obviously something that we do up front. Do we think the chemistry automation piece sits, most comfortably and makes the most contribution or most impact at those early stages as we're transforming hits from virtual screening or from high throughput screening into that sort of early lead expansion space? Perhaps Janeta, if you'd like to comment on that, and then Christian, maybe you have some views on this as well.
Yeah, absolutely. I have used various virtual screening techniques in my prior life. Not, let's say, we are going to do that exercise to rectify, but in previous companies in the hit identification stage. And then when you think about, parallel approaches and how that enables, let's say, not only hit finding, but hit to lead, that has been absolutely critical, because no matter where your hits come from, and the idea that being a virtual screen or that being any type of hit finding campaign, elaborating on those hits and figuring out which of those have traction and can be on their way to become potential leads, it's critical path. The more effectively that you can probe that and have confidence, then you have progressibility in your, let's say, campaign, the better you are. So this is where automation and thinking about how you build this targeted library of choice, where you, of course, execute on a specific hypothesis to building those targeted sets, the better you are. In my opinion, has been an absolutely key, intricate, let's say, feature of our strategy on how we, let's say, convince ourselves that we have confirmed hits on a path to lead optimization.
I see, thanks. And Christian, in your platform, can you talk a little bit about how you and your partners realize the real chemical materials that are the product of your searches?
Yeah, maybe to come back to the previous discussion point, I think the spot for automation is mostly at the very beginning. The speaker said it pretty clearly. Automation works best in a routine fashion where it is repetitive, always the same. The way we create our virtual chemical spaces is that we base them on routine, robust chemistries in order to increase the likelihood and the chances of success. So in a sense, we are trying to put out a fishing net as wide as possible and make that very first step very quick and efficient, so that we hopefully find some hits, and then later on in the process, it is always more and more a case-by-case thing, where the chemistry typically becomes more and more complicated, and you have to work with all sorts of tricks in order to get a particular modification on a compound in place and stuff like that. So I think automation will start, at least at the very beginning of the process, to make that more cost effective, more quicker, and productive overall.
Absolutely. So I'd like to think a little bit about this integration idea between virtual screening, route prediction, and the automation piece on the chemistry, and to understand how close to reality that is and what kind of impact that can potentially have. Dave, maybe you'd like to start a little discussion on this particular point. is this something that we can, foresee having an impact on our industry in the near future?
So thanks, Dave. I think this is an area which needs more focus at the moment, because I think it will have an impact. What we're effectively doing is we're drawing together the leading edge work that's going on at the moment in machine learning based approaches to reaction prediction, alongside the more rules based methods that are already out there, and combining that with what we know about the capabilities in terms of the types of syntheses.
Cursed by the internet connectivity problem.
That sounds like nobody can hear me. I can carry on talking, but I don't think anybody can hear me. I'm certainly not on mute. I'm not aware.
No, we can hear you, Dave. It's Dave Match.
I can hear you.
We can hear you loud and clear. I think Dave.
Okay. I'll start again. So I think this is an area which needs more focus going forward, because it's just starting to open up. And the reason it's opening up is because of the ongoing advances at the leading edge of synthesis prediction. And I think also a desire by people to be able to make the transition between effectively a predictive route and getting that into a laboratory environment where you can efficiently undertake that. There are some specific challenges, I think, in terms of automation, but also opportunities. The challenge, of course, is that you've got to be able to know the capabilities and the reagents that are readily accessible to you in an automated environment. The opportunity is the fact that once you've got all that in place, is you can make things happen very, very quickly. And I think there's also an interesting question to bring into this, the whole integrated approach, which is about the scale that you work out at as well. I've noted that there's a comment on the questions about being able to predict both synthesis routes, but also potential outcomes as well. And there's the recent publication from IBM, the Schwaller Group, looking at the prediction of reaction yields basically it's a deep learning type method. And I think what that's starting to bring together is that we can also now start saying, well, can we use something like that approach as a surrogate for the likelihood that a piece of chemistry is going to work? And that, again, that should help us in identifying those types of syntheses which are actually going to deliver molecules. Actually, whether it's whether they're made in parallel by scientists, or whether actually they're made in the automated manner. But I think this whole arena of combining more prediction with automation is going to become increasingly important. And one of the drivers for that, of course, is that you can then look at automating, algorithmic methods for optimization. So that can be trivial in terms of, well, actually, I need to make a molecule. If I haven't made it, I'm just going to try the next route on the list. Or it can be the more complex types of methods, which were showcased by what Andy Cooper's doing in Liverpool, and some of the process chemistry that's going on between the pharma industry and the groups in academia, like Abby Doyle, that are looking at effectively real-time optimization of synthesis in a fully automated or a semi-automated manner. So it's like it's certainly happening. I think the potential to extend what IBM is working on with one of the platform providers at the moment into more automated optimization is also going to continue to expand this type of space. So it's going to take a few years to bring it together, because I'm always over optimistic in these things. But I genuinely believe now that we have enough evidence out there that this is going to happen in a reasonable time frame. So 3 to 5 years. You're on mute, Dave.
I'm not quite sure why. Janeta or Christian, would you like to add any comment to Dave's feedback there?
For me personally, I haven't used any of the prediction tools in terms of synthesis, so I cannot offer any more insights here, but I would definitely, let's say, resonate with what Dave actually said in his seminar, is how, you choose your building blocks. That's important, and how you involve them, let's say, from the beginning in your designs, in your virtual screens. That will enable you to be more effective when you start building the molecules that you want, or the hits that emerge, from your virtual screen or any other screen. I think populating those, and that's what we've done in our own designs in terms of mining, the larger collections of available building blocks, which they are available in stock, right? And I know Le talked about their own collection of building blocks that are available. I think that makes synthesis a lot more effective.
Yeah, my comment would be that I see AI come into this process mostly in two places very prominently. One is as we automate or start to automate further and further this process, more and more compounds are actually synthesized. So we generate a lot of data about success rates of particular deemed robust reaction routes. And that should feed in well to future predictions of the synthetic success for other molecules that follow the same or very similar routes. So learning from what we synthesize today to better predict what we may be able to synthesize tomorrow. That is one aspect where I see potential great impact for AI methodology. And the second is in the overall process, which is typically the design, the synthesis, then the testing, followed by the next round of design. I think as we speed up this process, there will be more and more need along the discovery cycle to learn from the previous round, also in an automated, in an AI fashion, for what should best be selected and taken into the next round. So those, in my opinion, are the most prominent places where AI methodology and machine learning will have a big impact.
I'd like to turn a little bit to the library design piece, and maybe you can comment a little bit on the impact that having such a large building block collection available to you in doing library design and library planning has when you're at the stage early in a program of thinking about what compounds you're going to make, perhaps working with your collaborators.
Yes, the material. Yeah, could you hear me?
Sure.
Yeah. Yes. Yeah. I think the scaffold and the cost structures. Yeah. The reach of the materials was very important for the early stage research. Yeah. Actually, this cost structure will save help our customer. Yeah. Help our collaborators to research their structures and their patent. Yeah. For some very useful building blocks. Yeah. And to find either if it's an old drug or some new drug. Yeah. And their potential targeted position, and and I think we can do a lot of things with these structures. Yeah. And WuXi has a huge number, so it's provide a potential issue for us. Yeah.
to answer one of the specific questions posed to the team, do we share the structures of the building blocks and the templates that we make available for library synthesis? The answer to that question is yes, both as standalone collections, and of course, as we heard from Carl on day one and a little bit from Lee yesterday, we have a enumerated collection of those building blocks and core fragments built into a make-on-demand library. And this sort of feeds into a little bit some of the comments from Dave with respect to those scenarios where the chemistry, the products that are going into a virtual screen are encoded by a known synthetic route and available building blocks. This makes for a very rapid follow-up to a virtual screen or any other kind of selection process. And I think, always bearing in mind the availability of the tools and the speed with which you can follow up on a virtual screen with real chemistry is something that helps us accelerate programs, and very often helps our choice of chemistry and leads to follow up after a virtual screen. Simply the tractability, as Janeta mentioned a little earlier, the tractability is a very enabling aspect of creating more data that gives you more information that helps you refine your models.
Yeah, yes, as you know, in virtual screening, we will do some peptide protein optimization, and combine with our desired targets, for the Parry and reactor site, and after synthesis, and actually with after quick synthesis, we might find some hit, and if we can abate a crystal, co-crystal with our protein and our compound, which will help us to review our compounds, and it's a cycle to help us to find a better one.
Absolutely.
Actually, I think we have that successful precedence.
I'd like to go back to one of the topics I think Dave mentioned a little bit earlier with respect to other applications of automated chemistry and the potential impact in, for example, process optimization or reagent or catalyst selection. Dave, have you seen applications of that in manufacturing processes, and have some examples of the impact that that potentially has?
So it's always challenging when you're asked a direct question, because your mind tends to go blank. There's certainly a lot of interest in automation in the process chemistry world, but I'll be honest, my background is primarily discovery. But I think what we are seeing is that a lot of the commercial platforms that are out there are being utilized for process optimization, where large data sets are highly are both valuable and required. I think there's certainly been a couple of interesting examples that I've seen. One of which I can recall came out of the labs at Merck, where they were actually looking at not only potentially optimization of the chemistry in a very high density format, which they were able to achieve through automation, but also couple that directly with a relatively straightforward affinity based assay, which I think, if I remember correctly, was a mass spec based direct detection. So that's being done. There are other initiatives ongoing. There are various, obviously, the groups of Klaus Jensen, Avid Oil, etc., are very much focused on the leading edge of process chemistry type optimization, and they're certainly using a combination of sort of automation and deep learning techniques to speed up and potentially to do in real time the optimization process. I'm also aware of a number of academic groups that are active in this space, perhaps looking at real-time process optimization, particularly in flow, where you can have a very fast feedback of outcome type data from a reaction stream, and adjust the reactions, effectively make changes in real time to go through the optimization process. I'm trying to think, I'm not sure I know of any sort of examples where the impact has gone all the way through to the actual manufacturer. That's not to say that there aren't any.
Thanks, Dave. Thomas, hi, good to see you again. A question that maybe you're well positioned to answer with respect to the status of the algorithms for synthetic route prediction. We spoke about that a little bit yesterday, and the application of those tools as part of a selection process using the Schrödinger platform. Is this something that you think is becoming more mainstream and more widely available and used?
Not 100% sure about how widely available, but it is definitely something that everybody is focusing on. I mean, I think we are about to tie the last bit of the loop, and we can do a lot of calculations, make very nice predictions, and come up with cool molecules. But ultimately, you need to get somebody to make them for you. And as long as the automated synthesis robots are not at the stage where they can just do that, you have to convince somebody that that is actually a good idea. And synthesizability, to me, is an absolute critical component that I think the theoretical community does not do as well as it maybe should at the moment. So I expect certainly changes there in the near future, because that's one of the big remaining roadblocks, I think, for wide application of CompChem or integration.
Okay.
I think I can just come in on that, Thomas. The actual predictability of whether a reaction is going to work or not is a very interesting question. And of course, one of the challenges is that the data sets are not that strong in that respect. They're very good, or relatively good, because for successful chemistry, because that's what's reported in the literature, it's the lack of failed reactions, which is obviously a gap in the training set. And this is certainly an area where, within industry, and they have large collections of chemistry, both successful and unsuccessful, in their electronic laboratory notebook type records. They're in a good position to be able to make better use of synthesis prediction, outcome prediction, potentially yield prediction, because they have a much richer data set and aren't relying on effectively building a dummy negative set to help in training the algorithms.
And fully agreed. The literature is kind of blind in one direction, and that's a problem.
Yeah, but this data, if I can step in here, is slowly coming together. I mean, we have these make-on-demand compound collections now, and people are demanding, and not all make-on-demand compounds can actually be synthesized. And that's what I meant earlier. With every order, we definitely learn something from the synthesis, namely what works really well and also generalizes well, and what seems not to work well for whatever reason. And as I said earlier also, I think machine learning should be able to help understand the differences and then enable predictions of other building blocks that are amenable to a particular reaction or not. So Dave, my point of view on this synthesis planning is I always see it as you can come at the problem from two different angles. And either your design space is made such that you have high synthetic accessibility already integrated, embedded as the design rules. That is kind of the way that we code these chemical spaces that can be huge, as you have seen from my presentation. And the other side is you're moving off of that sort of grid of possibilities and do, let's say, more de novo style designs, then you need an ability to decompose your molecules and find ways to actually synthesize that compound that you have designed. We are very successful with Route 1, coding the space so that it covers likely synthetically accessible molecules. And I think the other way around is definitely more complicated. There is progress at that end as well, and maybe also machine learning is going to handle that part of the problem also better. But, well, at least you have two choices.
I can comment a little bit from our point of view with respect to the collation of negative data. Let's say one of the things that we've been doing really since the early days of library synthesis in WuXi is maintaining an annotated record of the reactivity of all the building blocks that we use in particular reaction situations. And you can think of that as a simple kind of traffic light system. It always works. Sometimes it works or it never works, kind of thing. But having that information available to you when you're doing library design means that in any particular situation, you can manage the distribution of highly likely versus highly unlikely products, and therefore make sure, particularly in the early stages where you're focused on data collection, really, you can focus your library on those molecules that you feel are much more likely to be delivered as a high proportion. And those that maybe you anticipate will contribute some critical data, maybe those are but are low likelihood. Maybe those are the ones that you take out of the automated platforms and you synthesize using more traditional routes. That's sort of a very pragmatic way of making use of the data. And as you say, Christian, we do generate a huge amount of data. And I think it is kind of incumbent upon us to learn as much as we can from that. But of course, an element of that is always needing to learn in a way that protects intellectual property. And I think this is one of the reasons why our data sets globally, in the industry, are very, very difficult to pool. So one of the problems that I think will eventually be solved, and in a way is getting there, perhaps indirectly through the synthesis route prediction piece, is finding a way to encode transformations that then take you away from the actual structure of the molecule, but only encode the transformation. And then you have data that you can't reverse engineer, which I think makes everybody a lot more comfortable.
Yeah, perfectly agreed. It's hard to say today how this particular field is commencing, but as you say, we have loads of data, and it's getting more and more as these methods, these make-on-demand molecules are synthesized, and not just a couple of hundred, but into the thousands. And over the years, we really accumulate a lot of knowledge, and we also try out more things, more off the main road kind of tracks. So things are certainly going to improve. And whether it's possible then to abstract from the exact structure and describe the molecules in a way that allows machine learning algorithms to benefit from data that is proprietary to any particular organization, generate a global overall better model, and then feed that back to the individual organization so that they can all benefit from it. I mean, there are initiatives like that already out there, trying to make descriptors and models work on, I don't know, global data sets, and then feed the models back. So nobody has ever shared their proprietary structures, to the other partners in a consortium, but all of them can benefit from the improved model. So something like that could work here as well.
I agree. I think, as I think we are generally becoming more collaborative across the industry anyway. So these kind of things are becoming more feasible. And as the tools become more sophisticated to allow that kind of reductionism to the reaction transformation, then I think this becomes possible. And the enrichment in the algorithms that result from that could be quite impactful. Let me just see if Thomas has any follow-up to that point.
I think Christian already summarized the main point well. I mean, all the data is there, and benefiting from it would be to everybody, what everybody should strive for, but the hurdles are big in doing it in a way that, let's say, a legal department would sign off. Obviously, this applies to a lot more things than synthesizability. I mean, you could think about tox models and ADME models and many other similar things where every pharma company sits on their own particular pile of data. And if you could pool all of these in a safe fashion, it would be immensely valuable to everybody.
So we're almost out of time, but I'd like to spend a few moments thinking about, postulating where we might be in a few years' time. So I'll give most of you the opportunity to think about that. But Janeta, I'll go to you first. If you'd like to comment on the discussion we've just had regarding data deconvolution in the synthesis setting, fantastic. But also, I'd like to think about where you think these aspects of chemistry automation are going to be in, let's say, 5 years' time.
Yeah, I think my takeaways, for example, especially in all the advances that have been done in virtual screening that, I took away from yesterday's seminar, that's very exciting. And I think how enabling these technologies are becoming to, let's say, to facilitate more effective drug discovery, it's been fantastic in the last several years. It's been, for me, it's been really, really neat to hear Dave talk about automation and where we think about it now, that automation has a great place on things that you are well worked out and know what to do, but also what the potential is as we go forward in the future. This couldn't be more exciting for a med chemist to think about how much more effective your optimizations are going to get. So I think, aside from just being humbled by the progress and internalizing and knowing how this would enable your teams to become more effective, that's fantastic to look forward.
Le, to talk to you briefly on this point about how you think automation is going to impact on your lab in Wuhan over the coming years. Do you foresee a time when you have mobile robots running around the lab and you have all the kinds of automation that Dave talked about?
Yeah, I think automation, it's a trend. Yeah, I think actually for us, we begin this test. Yeah, we use automatic machine for the library reaction setup. Yeah, actually, and it's beginning, and for example, and the flow chemistry are also utilized for some scale up. Yeah.
I think in your lab and indeed our other chemistry labs in China, that's going to be very impactful over the coming years. Yeah,
I think so. Yeah, but for the synthetic data, it's fun because IP issues. So I think we can't share it. So I'm not sure it could be into whether it could go into the big data.
No, absolutely. This is a point that needs to be solved by the industry to enable any kind of data sharing between partners, of course.
Yes, I agree. Yeah.
Christian, where do you think we'll be in a couple of years' time?
We as Biosolve IT obviously have a very clear path forward. I think in the past couple of years, we have seen a growth of chemical libraries from the millions to the billions. So in general, 3 orders of magnitude. And I think there is still a lot more room out there. We can show today already that working with trillions and even more molecules is feasible or will become feasible. On the chemistry side, on the wet lab side, this has to be supported in that we can actually not only design molecules in these huge spaces, but they can actually be made. And I think this is going to happen. And then we are in a situation where pretty much everybody will be used to take from millions, and no, not from millions anymore, from trillions of possible molecules, very, very valuable starting points. And then again, we as Biosolve IT are on the mission to make all the standard virtual screening technologies work on this kind of data and support the discovery process very effectively. So what I'm envisioning is an even shorter design cycle enabled this way. And then you put in AI methods into the equation. And I think that will do a lot to accelerate the early discovery process a great deal.
So Thomas, we're dependent on Schrodinger's platform taking our virtual screening hits, perhaps using FEP and a little bit of crystal structure data, and filtering those down to the 10 or 100 molecules we need to make to get to our preclinical candidate. When do you think we'll be able to do that?
I mean, I'd say it's a gradual process. I mean, having missed the talks today, and unfortunately, I have to take a very general take on that. But to me, I think the competition with automation, the robot labs, the flow chemistry is sometimes a little bit scary. I mean, I as a computational chemist, I mean, the machines are taking over should be my motto. But seeing that we as theoreticians, I mean, we have to keep running just to stay in place because the experimental side is getting so much better that it's the art is going to be to figure out where our methods need to be applied and where something else is a much more straightforward approach. And in for example, the DL plus enhancing the recovery of the compounds from that with AI, I think that I showed yesterday, that is hopefully is a nice example of where that niche could be, where you fit together the most modern experimental and theoretical approaches.
Perfect. Thanks very much. Dave, that just leaves you. Where do you think we'll be in terms of automation in a few years' time?
So I would echo some of the comments that Christian made. I think that's definitely all going to be very, very feasible. I think what I'd like to see happen is twofold. I think the first is to ensure that we always work as efficiently as we can. And automation is just a tool to drive us towards a more efficient process. And so it's not just about the individual steps, it's actually about bringing those together. And so if you look at the entire process of discovery, the design, make, test cycle, I think we have to come back and we have to look some more at improving the integration between the synthesis, purification, and through to at least some primary assay data. This has been demonstrated by a number of groups, cyclofluidic, AbbVie, AZ come to mind. I think in what they have demonstrated is the potential to be able to in some way close the loop. So we can go back to what Christian described a little bit earlier in terms of being able to be much more efficient in the optimization of those early stage hits through to molecules that we have confidence can be progressed. I suspect we're a little way away from just being able to make a couple of dozen compounds to be able to get a clinical candidate. But I think, you know, there's a lot of streams coming together at the moment, and I would hope that in the 3 to 5 year time scale, we will see that, you know, automation is much more routine and is delivering to be, you know, a core part of the discovery process, just as virtual screening has become over the last decade or so.
Thanks a lot, Dave, and thanks everyone for the discussion and Q&A. And thanks, Lua and Dave, for your presentations today as well. And as well as the previous speakers, Christian and Thomas on the call now, but also Lee and Carl from earlier in the week. I'd like to close by thanking my colleague, Nick Foster, for doing the main legwork on coordinating and setting up this discussion, and also the organizing group, LD organization, and Lai and Bruin in particular for their coordination. If you do have any follow-up questions, please do feel free to reach out to me. And of course, I'll be more than happy to connect you with any of the speakers. Very, very grateful for everybody's time contributing to this symposium. I hope everybody enjoyed it, and you will in due course get a link to the recording and copies of the slides. And I'd like to close by encouraging you to complete the survey that will be part of an email link to you, and will also be on your screen shortly. Thank you very much for everybody's contributions. Goodbye.
Thanks, Dave. Great meeting.
Thanks very much.
Bye-bye, everyone.
Bye-bye.